But since you're saying it, that technically means that you just gave us an extension on this thing if we watch the movie.
I think you can either or. So you can watch the movie and write me an editorial on how the Reanimator is terrible science. Or you can submit the ethics proposal that you're probably already working on.
Joke's on you. So can I do this first one?
Guarantee it'll take longer, but if you really want to, I will accept it. I did actually used to have an assignment. I had 2 editorials in this class before. One was watch a movie or an episode of something and then analyze the genetic material, whether it's good or bad. People didn't seem that excited about it, so I can do it again.
I don't know, right?
It's like I'm forcing you to watch Netflix. I'm sorry. All right. So, okay. That is a nice lead-in to the fact that your ethics editorial is due on Friday. I've got a few questions on those, so if I haven't responded, I will. Are there any questions I can answer now? Remember, they're not terribly long. You're choosing a side. I don't care what side you choose as long as you defend yes.
When you say like references are not included in the word count, do you mean like our parenthetical inserts and stuff? Or
No,
you just
that
mean, like,
would be
the
included, but I
sources
mean your
that-
like, yeah, your actual references,
Okay.
your list cited would not be included. Because that could be 500 words on its own.
I was gonna say,
Yes.
like we can't.
Yeah. Other questions?
So you can't use quotes either?
Yeah, it says
Like in
quote
one
should
part?
not be used, and then one part it says that they can.
Yeah.
Oh, sorry. So I prefer that quotes not be used. If there is something that you really, really, really want to quote, just make sure it's a short quote. So what I don't want is like 250 words of a quote.
I'm using 4 block quotes. I'm just kidding.
The whole interview.
And then so-and-so said 250 words, and so-and-so replied. That's what I don't want. So I prefer you avoid quotes,
Use block.
but if you do include them, make sure they are particularly relevant and short. Fair? I apologize for the disagreement of my own assignment. All right, so that's due Friday. I believe I have the Dropbox set at midnight. If it's not set at midnight, just submit through midnight. I'm not going to grade them Friday at midnight, so as long as they're there before I see them, before I download it all or check it out. Okay, also due Friday is Chapter 8's worksheet. Yes? Hopefully people have been, I know some people have been working on those because I've had questions. I will give a little bit of time on Friday at the end of lecture to sort of go through anything that we talked about on there. What else? I think lab went pretty well on Tuesday, so I expect it'll go swimmingly again tomorrow. Don't forget, if you're in Thursday's lab, to bring safety glasses or goggles. And I think that's really also wear closed-toe shoes and so on and so forth. Our next exam is in 2 weeks. Oh, guys are so grumbly. All right. I gave you one of them a month ago. And one here.
And there was a test in there too.
Okay. All right, so we are going to finish up, or I'm just going to revisit this last little piece of Chapter 8, and then we'll go into Chapter 9, so we'll start DNA replication. When we talk about repetitive sequences in a chromosome, we've already actually talked about one situation where they can cause issues. Does anybody remember? Actually, we've talked about a couple. When repetitive sequences may actually be sort of problematic or can complicate processes that we've already talked about. Yeah.
It can cause them to like misalign when they're matching together.
Yes, exactly. So when our chromosomes line up during meiosis, they line up based on sequence similarity. A lot of these repetitive sequences are sort of long stretches of repeat information, hence their name, that's inserted at one location, accidentally duplicates itself and inserts at a different location. That can cause chromosomes to misalign, and then we get unequal crossing over, so we get deletions from some chromosomes and duplications in others. Right? So that is sort of one area where we've talked about these. They can cause other issues as well. So within a genome, we have sort of 3 levels of sequences. We have sequences that are unique. Those would be our gene coding sequences primarily, right? Every one of our genes codes for a unique product, so a unique protein, or a unique RNA, like a tRNA molecule. If we have moderately repetitive sequences, those are sequences that repeat a couple hundred to 1,000 times, or a couple thousand times throughout a genome. And then we have our highly repetitive sequences, and our highly repetitive sequences are found thousands and thousands to even millions of times throughout a genome, and I think it's down here. Yeah, so under highly repetitive sequences, we actually have a family of non-coding sequences that we call the ALU, or the ALU family. This particular sequence is about 300 nucleotides in length, and it makes up almost 10% of the total human genome. Okay, so that 300 nucleotide repeat segment is so common in our genome that it makes up about 10%. Right? We're talking about 3.2 billion nucleotides or base pair nucleotide sequence. So 10% of that is a significant number of these repeat units. I'm not good at math, but 3.2 billion divided by times 10%. Where's that? 300 and, I guess. It's a lot. Yeah. So essentially what that means is if you were to look at sort of a chromosome or a string of DNA, every 5,000 to 6,000 base pair, you would find that repeat sequence. So it'd be like ALU sequence, a couple thousand base pair, ALU sequence, a couple thousand base pair, right? So it's inserted sort of over and over and over again throughout our genome. Most of these highly repetitive sequences have been inserted historically by viruses, right? So we do have actually quite a few viral sequences in our genome. They don't really code for viruses anymore. Do you guys know how viruses replicate? Yeah.
Is it through binary fusion?
So that's that's bacteria.
Oh, okay.
Yeah. You're not entirely wrong though. Where do you guys learn about virus and bacterial replication? Do you talk about it, Micro? I used to cover it in here, but it was kind of out of place.
Yeah, a lot of us don't need microbiology for forensic science, so.
Right. Yeah, so there's actually quite a few different types of viruses, but some of them, what they do is they inject their DNA into a host cell. The host cell then identifies that as a piece of DNA that needs replicated, and so the host cell's machinery actually replicates the viral DNA. Eventually, that causes the cell to rupture and release all of these viral DNA or RNA pieces. Sometimes they get inserted into the host genome, and then the host, so that's what with bacteriophages, they insert into the genome entirely, and then the bacterial cell replicates the genome, boom, you get viruses. So sometimes when those viral sequences get inserted into our genome, they then sort of mutate, and they don't make viruses anymore, but we sort of have this now leftover piece of DNA that didn't belong there. All right. Yeah, so we just looked at this quickly at the end of class, where within the human genome, the percentage of our genome that's actually unique sequences, so genes that encode proteins, gene enhancers, so I think I call these promoters and the like initiators, things like that. Transcription factors. So these are outside of the gene coding region, but they actually serve as sort of signals. So they signal molecules and enzymes that are needed to start a process. And they say start here, and then so that they know sort of where to land and begin that transcription process. And then other unique sequences that are not gene coding are also part of our genome. But repetitive, so mildly and highly repetitive sequences make up almost 60% of our genome. So that math is a little bit easier to do. That's over 1.6 billion of our nucleotide sequence in our chromosomes is not used specifically for coding genes. All right. Replication. Any questions? I was trying to look for a really cool replication video this morning, and I failed miserably. There's lots of really terrible rap parodies for replication. No, they were not worth it. I feel like I wasted half an hour this morning of my life listening to high school teachers rap about DNA replication. Great, not good. It's terrible. And then I got sidetracked even more and started looking up. Have you heard of Slash Street Boys?
No.
They parody, so they're a Halloween cover band, but they parody Backstreet Boys songs. So that's another 20 minutes of my life. Okay, so for DNA replication, we have quite a few objectives. Yeah, look at those, so nice. So the first thing that we're going to do is actually go back to the structural features of DNA. So we are going to think, pair, share our what we remember about DNA structure, and we'll talk about then how that structure helps us start this process and actually even makes this process possible at all. And then again, we're going to go to the bacterial side of the world and talk about bacterial replication. At the end, we will start throwing in a little bit about eukaryotes, but this is where sort of our understanding of the eukaryotic process becomes a bit fuzzier. So we aren't entirely sure all the steps that are involved here. And then everything else is just what we're going to talk about in between those things. All right, so I want to start off just by giving us some numbers. It's not math, well, it's kind of math, just numbers in general. So if our genome is 3.2 billion nucleotides in length, that actually means that we have a total of 6.4 billion base pair. When we give this number, we're talking about our double-stranded helices. Okay. So we're talking about it as two parallel strands. Here, this would be if we uncoupled the double strands, we now basically double the number of nucleotides that we're talking about. Does that make sense? Right, so here they're paired in their complementary strands. Here is if we pulled them apart and considered each base pair or each nucleotide on its own. Okay. So when we go through DNA replication, we are actually replicating this number because we are taking our DNA strands and we are pulling them apart, and we're using each side of that chromosome or each rung of that ladder as a template for a new strand of DNA. Okay. So when we are replicating this 6.4 billion nucleotides, if we had an error rate of 1 in every 1 million nucleotides, how many errors would occur during that replication? Well, first let's start here. Does 1 in 1 million seem like a very high error rate? No, it does not seem that high, right? I mean, that seems like a pretty low error rate if we think about it. But if we're copying 6.4 billion base pair every time we're replicating our genome, so when we're going through that S phase and interphase of cell division, we are taking 6.4 billion nucleotides and we're copying them. So if we have an error rate in 1 in 1 million, how many errors would occur during that single replication? You're off by 1 factor.
That one doesn't go
Oh, do I have it? Is it on my slides, Ron?
Yes.
I didn't even know it was on my slides. No, it's not.
You're
600?
missing a zero.
Oh, in the million? Oh, okay. Fair enough. All right, there we go. Okay. So we would get 6,400 errors in just that one round of replication in one cell. So when we consider how many cells are in the body and how often those cells divide, how many cells are in the body? Say that again? A lot, like trillions, like 3 trillion cells or so make up the human body. They are constantly replicating, or we are constantly going through meiosis. In either case, if we have an error rate of 6,400 errors per replication, how does that end up looking for our cells? Yeah, not great. You probably don't want 6,400 errors in your genome every time it replicates. So we actually have an error rate much, much lower than 1 in 1 million. So the point sort of being is that as we go through DNA replication, one thing that we'll talk about is the fact that DNA replication is incredibly faithful and is incredibly reliable in terms of making exact copies. So it is actually very rare that we end up with errors. If we do have errors during DNA replication, we also have some processes that we can use to go back and correct those errors. So yeah, it's really important that we end up having complete, perfect copies of our DNA. Because if not, we end up with mutations and proteins that don't work, and it's not so great. All right, the other really important thing, or sort of interesting concept to think about if we're thinking about numbers, is in E. coli, they have a genome that is 4.6 million base pairs in length. So it's much shorter than our chromosomes or our genome. If they copied at a rate of, is this on a slide somewhere? I don't think so. Of 1,000 nucleotides per minute, it would take them 3 days to replicate their genome. How quick do bacteria replicate and divide? Say that again? Yeah, it's like 20 minutes is usually about how long it takes E. coli to replicate and divide. So they are actually replicating at a rate of about 1,000 nucleotides per second, which gives them about 20 minutes to replicate their chromosome. So not only is this process really, really good at being error-free or having very, very, very low error rates, but it also happens super fast. 1,000 nucleotides a second, that's insane, right? And in an E. coli, I think this is E. coli, which we know that E. coli's chromosomes look much different than ours, but we're talking 4.6 million. Humans, we're in the billions. So it does take us longer to replicate our genome than it does take a bacteria to replicate their genome, but it's still not days or weeks. So our cellular machinery also replicates very, very rapidly. So So it's actually a pretty impressive process if you think about it. All right. So what we're going to do today is essentially talk about how, or sort of the overall model of replication, and then I suspect on Friday we'll get into which enzymes are involved and all the pieces sort of fall into place. So before we do that, I am going to tell you to take 1 minute, don't use your notes, take 1 minute to think about the structure of DNA on your own. Then I'm going to have you make a list of all of the structural things that you remember about DNA with your neighbors, and then we'll go through it as a class. So take a minute to think about what you remember about DNA structure. Think of it as a pre-test. Yes, I have one structural component written on the board. It's over here. All right, take a few minutes to share with your neighbors. Come up with a list as comprehensive as you can get it. Yeah, actually, I had to do that for like a My husband has that built in as extra credits one of his courses, but
Yeah, we just like rap. I wanna do is
super stringent on the, like it has to be.
Has to be like legit.
Yeah. I would help him memorize that. We sit there like.
Hair? Jesus, cheese. I can't have it right now.
Oh, that's yeah.
How many markers have you had to throw away already this semester?
A bunch. I write a lot more than some of my other classes though. Also, these are like really year old. You guys ready? You guys have comprehensive lists of DNA structure? Yes. All right. Let's hear it.
Phosphate backbone.
Maybe like to add that.
Sure.
Yeah, thank you.
Yeah.
Okay. Yes?
Yeah.
Good start.
Thanks.
What else?
There's another.
Thank you. A and C has 2 hydrogen bonds.
And
Oh,
C to G has
So, okay, so you are talking about what property though?
base.
What about base pairs? Base pair pairing, and they pair in a,
What way?
Pairing
through
hydrogen.
yes, say that again?
That pairing,
Nope.
the pairing.
Not what I thought I heard. Okay, so we have complementary pairings, which are going to be A and T, oops, and C and G. These and these are hydrogen bonds. Yes? Okay. Let me ask you a question. What type of bonds hold our phosphate sugar backbone together? Phosphodiester.
Good job. How did you guess that? Okay.
And if we had to categorize these bonds into our broader categories of ionic, covalent, hydrogen, covalent, yep. These are covalent bonds. Okay. That becomes important in replication. Which of these bond types is stronger? Which of these bond types is stronger? Covalent. Yeah, hydrogen bonds are little weenie bonds. They're easy to break. They're strong in number, but they're weak in sort of individual pairs. All right, what else? Okay, what do we call that? Anti-parallel.
I know the base.
Oh, parallel. Anti-parallel strands. Okay, and so that means that we have one strand that runs 5' to 3'. Its complementary strand runs this way. Yes? How do we know which strand runs 5' to 3' and which strand runs 3' to 5'? What does the 3' and 5' mean? The carbons, right? We number the carbons in our sugars. 5' carbon on top, 5' to 3'. They flip over in the opposite direction. 3' to 5'. What else?
A and G are pairings.
Okay, so we have, kind of try and throw that under our complementary base pairing. So we have purines, which are going to be adenine or adenine and guanine. And then we have, oops, thymines, which are thymine. Might as well for me, I'm not sure. Okay. Other features?
It's a double helix.
Thank you. Whew. Yes, so we have a double helix structure.
It's right-handed.
It is right-handed.
The major and minor groove.
Yep, so when we get this double helix, sort of along it we have major and minor grooves. What do those grooves do? What do they allow for?
Access to nucleotides.
access to the nucleotides. Which one has more access to the nucleotides? Major. There's more space there. Yes? What else do we know about our double helix, right-handed, twisty ladder?
Oh, how many base pairs? It's 10 base pairs.
Yep.
Turn.
So we have 10 nucleotides per 360-degree turn or twist.
Each base pair is 2 millimeters apart?
3.2.
Okay.
3.2? I think it's 3 point something. Yeah, so if you get a length, 320 nanometers, you could infer that that is 10 360-degree twists or turns. Let's see. Any other pieces? Yes, so our nucleotides are hydrophobic. And that is important because it plays a role in the stability of DNA. So it keeps, remember that your cells are just bags of water, and so we have all this water, it's going to push those hydrophobic pieces together, right? So it pushes the nucleotides into one area. We add that to our hydrogen bonds that occur between them, and we have a nice strong force that keeps that those complementary pairs really tight. There's one more thing sort of about the nucleotides that's important. So the other sort of piece that's important about the nucleotides is how they stack. Remember, they stack flat and not vertical to each other. And again, that is important in sort of stability and access to those nucleotides. Does anybody have anything else for this list?
Is deoxyribose.
Okay, so our sugar in this case is deoxyribose. So literally everything that we've listed up on that board plays an important role in the error rate, or the low error rate, and the rapid replication rate of DNA. So those features are super important to this process. All right, so we're going to take the next 10 minutes and we are going to sort of talk about different models for DNA replication. So again, we're taking our step back to the 1950s and 1960s when we knew what DNA was at this point, we knew what chromosomes were, we knew meiosis and mitosis, but we still weren't sure what this actual process looked like. So, we sort of have 3 different models that were proposed or examined. The first one is our conservative replication model. In conservative replication, we would assume that the parental strands, and when I say parental, I mean sort of the original piece of DNA or the original strands of DNA, separate, are used as a template to make 2 new strands. Those strands then separate, and the new strands come together to form a molecule, and then the old strands come back together and form a molecule. Okay, so we would have our original DNA. This DNA molecule would separate into 2 pieces. Right, so it would separate into this side and this side. Each of these would then form the template for a new strand. So a new strand would be shown in red. Try it. No. technology, it'll be great. So this is our new strand. New strand. What would happen next is these two would come apart and these two would come apart. These two would then join together to form this molecule, and then our original strands go back into sort of their original conformation. How does that sound to everyone?
Weird.
Maybe like some extra steps, right? Our cells are pretty efficient, so maybe this doesn't make the most sense. Is there seriously a red dot here now? What? Just not when I write with the marker. Just use my finger. All right, so the next model that was proposed was the dispersive model. In this case, we have sort of equal representations of parental or old or original DNA and new DNA, but they don't stay together in strands. So in this case, I'm not even sure how I would draw this out, but these would separate and then break into pieces and then come back together in the new molecules. So we would have these molecules where we'd have basically two complementary pieces that were original, and then two complementary pieces that were new, and then two, right? So we kind of string it together so that it ends up sort of broken up into these chunks of original and new double helix. All right, and then our third model is our semi-conservative model. So in the semi-conservative model, we start off again with our original strand of DNA. It separates, and so we get our template strands on one side and our template strand on the other side. We then get our new DNA, right? That basically copies or is complementary to the old strands. These two then double helix up and form our daughter molecules. What do you guys think? Okay. Yeah, so semi-conservative is probably what makes the most sense, just in terms of sort of ease or how simple, not the simple process, but that is going to be more simple than breaking apart, making new, breaking apart, remerging. Most efficient. Most efficient, yes. So the experiment that was done to sort of verify this was done by Messelson and Stahl. This was in 1958. So that seems like a long time ago, but not too long ago. Their hypothesis was that DNA replication followed that semi-conservative model. Watson and Crick were coming out with their understanding of DNA structure just slightly before this, 1953. And so they did an experiment where they tested this. Okay. So what they did was they labeled nitrogen. Nitrogen is necessary for production of nucleotides, right? So our nucleotide bases have nitrogen in them. So they labeled nitrogen as heavy nitrogen, so it was nitrogen 15. Into that nitrogen 15, they put E. coli, and they let the E. coli stay in that nitrogen 15 environment for quite a few generations. So what that means is that those E. coli, as they went through mitosis and binary fission to make new individuals, they used heavy nitrogen to make their nucleotides. Okay, so they take nitrogen from the environment, they incorporate it into their nucleotides, and then they can replicate their DNA. So after they had grown E. coli in that nitrogen 15 media, they moved them into a light nitrogen or a nitrogen 14 media. And they let them stay there for one generation. So the idea now is that we've changed to a different type of nitrogen, which is a different isotope. It leaves a different signature when we're analyzing, right? So we can see heavy bands and light bands. After one round of DNA replication, right? So their parent strand has only nitrogen 15. So it has all heavy nitrogen. Once they're in the light nitrogen media and they make a new strand of DNA, it will have only light nitrogen. So if you take the DNA out, what would the ratio of light nitrogen to heavy nitrogen be after one round of replication? Okay, so this molecule is all heavy nitrogen. Every nucleotide in here is made with nitrogen 15. Each of these red molecules would be made with the new nitrogen, which would be nitrogen 14. So what is, and this is just one round of replication, so what is the ratio of blue, or nitrogen 15, to red, or nitrogen 14 in this generation? 50-50, right? It's a 1-to-1 ratio. So if they took the E. coli after one generation and they separated heavy nitrogen from light nitrogen, if we do in fact see semi-conservative replication, we should get 50-50. What about here? What would the ratio be? 50-50. Or here? Yeah, so the first generation doesn't really tell us much because in each of these we would expect half of the DNA to be light and half of it to be heavy. So they kept going a few generations, and eventually what they find is that we end up with sort of an intermediate. Okay. So in the intermediate, what that means is that we have one strand of old or original nitrogen 15 DNA matched with a strand of new or light nitrogen DNA. Okay, and so that actually means this isn't true, and it means this isn't true. Okay, so in this case, we would end up with lots of light nitrogen strands and then one heavy nitrogen strand. Here, all of them would actually migrate to the same point because they have an equal amount of light and heavy. And then here, we would have two strands that would be kind of intermediate because they have some light and some heavy, and then two strands that are all light. Okay? And so that's what their results actually show, is that as generations proceed, we end up with more, and actually this is going this direction, so these are the first generation, and these would be further generations. We see that we end up with more light nitrogen, but we still have kind of this faint band down here, which is like those original two strands of heavy nitrogen that stay in the cell. They just become proportionately less than what we see of the light nitrogens. So, you can watch a little video.
For semi-conservative replication of the DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contain heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density.
So that'd be two light strands
DNA
together.
containing N15 is denser than that
Original
containing
strands.
N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Messelson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
Excellent. And there we have our introduction to DNA replication. So on Friday, we will talk about how this process actually happens at the nucleotide end. So I hope you're excited. I hope you enjoy your Halloween. Sorry. Sleeping? Me too. Actually, I have to know. I'll see some of you in lab tomorrow. Otherwise, enjoy your next few days. You'll be okay. It's practice.
No, it weighs 6 billion pounds. I'm not gonna watch it. One time I had buffalo sauce there, it exploded, and then it smelled like buffalo sauce. I was like, why does it smell like buffalo sauce? And it really stood because it exploded. I had to watch it. And Dower cheered up once, I remember so with that.
Oh, you know
This is a little
We're looking for you.
Why am I a cat person? You have a shoe bag.
Actually, my cats chew my backpack more than my dogs.
You wanna go shower? What are you doing?
Yep, that's the drip water.
Go somewhere.
I'm ready to go back to bed.
Yes, it's a different color.
Well, I can go home after this.
No, I totally thought today was Thursday. Okay, how do you just like quick research?
Yes, please, if you don't mind.
Jordan. Yeah. Yeah.
Everybody's getting one.
Oh, wait, how do you make one copy?
Yeah, we do copies. Just do like 25 or something like that. Hopefully that will be enough.
Why does it say you make even copies?
Is this 18 million?